morning, ladies and gentlemen. Assalamualaikum warahmatullahi wabarakatuh. It is a great pleasure to welcome you to this webinar on the 13th Cryogenic Electron Microcopy with topic protein purification, crystallization, and structural analysis of novel case VAPD subfamily protein Burkholderia Pseudomallei, held by the Directorate of Laboratory Management, Research Facilities and Science and Technology Area BRIN, today Tuesday, December 10, 2024. In this occasion, we are honored to welcome Dr. Ing R.B. Dimyati, Researcher at Research Center for Nuclear Beam Analysis Technology and Coordinator of Cryo-EM Laboratory BRIN. Our speaker, Professor Long from Institute of System Biology, University Kebangsaan Malaysia, and our moderator for today, Dr. Dede Heri Yulianto, researcher at Research Center for Applied Microbiology and manager of Cryo-EM Laboratory BRIN. Okay, ladies and gentlemen, next agenda is singing the national anthem Indonesia Raya. Ladies and gentlemen, we will move to the next agenda. Is opening remarks from Coordinator of Cryo-EM Laboratory BRIN to Dr. Ing R.B. Dimyati. Time is yours.
Thank you very much, everybody. Can you hear me?
Yes, quite clearly.
Clear.
Yes.
Thank you. Good morning, everyone. So it is my pleasure to have you join us today for the second protein purification webinar of our team. I want to extend a warm welcome to all of you, whether you are joining us from near or far. Today, we are here to discuss technology of protein purification. As many of you know, we are lack of purification unit, and we have now the most modern purification unit here in Indonesia, located here in Cryo-EM lab in Cibinong. So I am excited, or we are excited to have an expert presenter, Professor Qian Leong, who will share his insight and experience with us. So we are sure so his insight will be invaluable to all of you. So I would like to express my thanks, especially for the organizer under lead of Professor Dede, uh, under whom it is this webinar is possible. So I would like to open this session officially. So I wish there is a good discussion. So thank you very much. And the floor is back to moderator. Thank you.
Thank you, Dr. Abdi. Ladies and gentlemen, before we start the agenda, we will have a photo session first. To all participants, please activate your camera and for the committee to take the screenshot. Okay, ready? I will start count from first slide. 1, 2, 3. Second slide. 1, 2, 3. Thank you. Ladies and gentlemen, without further ado, now we are going to start our discussion for today, which will be led by Dr. Dede Heri Yulianto. So, Dr. Dede, time is yours.
Okay, thank you very much, Bu Yeti, and also Dr. Abi. Ladies and gentlemen, this morning we have a very good Zoom seminar with our speaker, Professor Leong. Thank you so much for your time to join and as a speaker in this Zoom meeting. We have a good communication with Professor Leong from University Kebangsaan Malaysia. And I think it is a good chance for all of us here to learn how we purify and crystallization and also structure determination of the novel subfamily protein from the Burkholderia Pseudomallei. I read this paper in Nature. It is very good paper from Professor Leong and team. In this session, he will share to us how he purified, how he purification, crystallization, and how the structure determination of this novel protein from the bacteria is resistant to antibiotic. Yeah. I think it is very good for us. Professor Leong is a professor in the University Kebangsaan Malaysia, Institute of System Biology. Thank you very much, and time is yours, Professor.
Thank you. Thank you. Salam sejahtera. Selamat pagi. Good morning to everyone. Thank you, Dr. Abi, for your kind words just now. Thank you very much, Prof. Dede, for your invitation, for giving me this opportunity to share some of our works. So I think I would like to start sharing my slide.
Yes, please.
If this is okay. Can you all see my slides?
Yes.
All right. Thank you, Prof. Dede. So as Prof. Dede had mentioned, we have a very good discussion regarding if how our collaboration in the coming future regard about the protein structure or functional study. So with that, then I thank you so much for the invitation again for allow me to share some of our works that related to protein purification, crystallization, and analysis of hypothetical protein. In this case, it a hypothetical protein from Burkholderia Pseudomallei, which after we solve a structure and we found out that it is actually belong to a class 2 VAPD subfamily protein. So also from the discussion with Prof. Dede, where I knew that the protein purification and crystallization may be an interest of audience, so I included some slide of purification and crystallization for protein in general. So for many of you, if you haven't been to University Kebangsaan, Malaysia, or the National University of Malaysia, here's our main campus in Bangi, Selangor, Malaysia. If you look at the map, can you see my mouse cursor?
Yes, yes, yes.
Can, yeah?
See your
So we
cursor.
are about 30-40 kilometers from Kuala Lumpur and near to the Putrajaya and also not far from the airport. And I'm now currently in Institute of Systems Biology, which is established 2005. And we are interested in tropical biodiversities and also sustainability for the discovery of new genes and also biomolecules. So we also apply transgenic for crop improvement, also synthetic and systems biology to understand holistically how a cell works as a whole. And there are several focus that the Institute is focusing biotechnological applications of local biodiversity. If you can see that many plants that you may be familiar with as well, venturing into a sustainable bioproducts. Also trying to contribute for the food security issue that or food security issue that the world is actually facing. And we apply omics technology in order to come into solving the problem or challenge that we are facing and understand the system biology as a whole. And I ultimately, perhaps our finding can move to a translation and research that will be benefit the economy and the nations. So today, I'm for my works, more focusing on structural biology, which we can understand the structure and those function that allow us to know a pathway or function of protein or enzyme. And these showing a few structures or the structures that we have solved in Institute of Systems Biology for the past 10-12 years using X-ray crystallography. So we have solved a structure like from human, plant, plasmid, or even hydra for the eukaryotic, and few structure from Burkholderia, Pseudomallei, and also different bacteria. As from the pipeline of X-ray crystallography, we are involving in producing or cloning the genes and then towards getting the protein until solving the structure. So you may see a couple of time of this slide because there are several parts that I would get into a bit more detail. Is especially from protein purification facilities over here. So just for your information, over here, in order, as I mentioned, we solve several structures in Basis. So most of the structure that was solved with the method as follow, where normally we are interested in certain genes. So in the past, we may use a PCR to PCR out or the genes and clone into the E. coli. In our case, we are mostly using E. coli because it's most simple and fast. So we will clone into the E. coli expressions vector. And with that, we'll transform into E. coli expression host, such as BL21 DE3 cells. And from the transformation, we are using an antibiotic selection to select those transformants. With those that are known to be positive or successfully transformed, we will then isolate and send for sequencing to make sure that the genes that we clone is correct without mutation. So after we have obtained the clone, we will then move into small scale expression. Small scales means 5 ml to 20 ml perhaps, or even you can do 2 ml, some people. For just a quick expressions test screening with a 16 degree and 37 degree in general. So that's a two-step for us to screen the protein expression to see whether the E. coli expression host can express the recombinant proteins. Once we obtain the clone that able to express the recombinant protein, and particularly soluble, because some protein can be expressed but not soluble. So once we obtain that, then from the small scale, a few ml, that we will then move to producing it into 1 liter or 2 liter culture in order to get enough protein for crystallization. So in 2 liter or 1 liter, we will use like 2 liter flask. So in this case, then you may use 1 or 2 or few to grow the cell, and we normally induce as log phase, which is about 0.5, 0.6 optical density of the cells. After that, after we harvest the cell, then we will suspend it in the buffer. So the buffer in general would be the buffer that we think the protein would be happy with, which means that we hope that the recombinant protein in this case would be soluble. And we will then lyse it using a sonicator. And after we lyse the cell lysate will be centrifuge, and the supernatant will then be subjected to purification. In this case, we are using two-step purifications, so which is affinities and side exclusion chromatography. Sometimes we will also use ion exchange. So it depends on the protein, the purity, and the type of protein. But most of the time we start with these two steps purification. So for the affinity, I forgot to mention, during the recombinant protein construction, we usually put a heat stack at the N terminal or C terminal, so that we can use nickel NDA or nickel affinity column that could bind the protein with a fusion tag, a 6-time heat stack. And from first purification, we will then harvest the peak of the protein and subject to size exclusion chromatography. And we have 2 types of columns over here. So it depends on the size of the protein, whether we will use like column that can select smaller protein from below 100 or bigger than 100 kilodalton. So after we obtain the purified protein, we will then use the protein subjecting for crystallization. So in the crystallization, normally we use 96 well sitting drop plate for the screening. And for the screening, we will then use a commercial available screening kit, which is crystallization screening kit, where they provide, there are several type or various type of the crystallization kits. And for the 96 well screening, we only need a small amount of protein. Since we are using manual setting of the crystallization screening, the smallest we can go is 0.5 microliter. Sometimes if we are lucky to have more protein, we could also use actually 1 microliter to 1 microliter, 1 microliter of the protein to 1 microliter of reservoir, means that we will mix 1 + 1, so there's 2 microliter in a drop. Or if we don't have many protein, some protein is very hard to purify it, then we will put 0.5 to 0.5. The ratio is 1:1. And with that, I think it take about 1 hour to set up one tray because we don't have the machine. If there are many proteins need to be set up, that will be nice to have machine since we don't have large amount of proteins. So this method is still workable. The structures that I show you where we have solved here are mostly using this method. And once we get the crystal heat over here, we will then optimize the conditions that we knew from the commercial available data sheet, because each of the condition have a different compositions of chemicals or precipitants for or we call the condition that would crystallize the protein. So by changing or varying the composition, concentration of the composition of the chemicals in the crystallization condition, we can then using or optimizing the crystal to, in this case, normally we want to have it bigger crystal so that we could have a single crystal that suitable for X-ray diffraction data collection or screening. So if it's for micro ED, of the day I learned that from the last week conference in the Asia Crystallography conference, that for micro ED probably people are looking for small crystal, small enough that it's not too big that or the electron can pass through.
Hello, Prof. Leo? Okay, I think there's something problem with the connection for Leo, and we will wait for him. Yeah, he just mentioned about the micro ED. So micro ED no need the big crystal. We just only need very small crystal below 0.2 nanometer the size, and we can use micro ED for the factor determination for the low molecular size below 80 kilodalton. Okay, I will contact Professor Leo.
Sorry, my apology. It's disconnected.
Okay, yes, please, Prof. You can share again.
Yeah. Can you see the slide?
Is it okay, the connection now? Yeah.
All right.
Yeah.
Sorry.
Yeah, you,
my
from the micro ED, the crystal size, I think you stop there.
yeah. So I learned that for micro ED, instead of need a big crystal for X-ray, but micro ED need a tiny crystal that allow the electron to pass through. So I think it's kind of a reverse of technique. But in order to grow the crystal, so we will incubate the plates in a chamber. For example, this is just a normal refrigerator that people sell food, and we set it to about 20 degree or 90 degree so that the temperature is stable. And in order to grow the crystal also, we move actually the compressor out so that the refrigerator will not be vibrate, because vibration is not good for growing the crystal. So that was I learned from a friend of mine, Dr. Ho Kok Lien from UPM. University Putra, Malaysia. He showed me that how he set up the crystallization this chamber. So I learned from him to move out the compressor so that we have a stable environment that we're able to grow the crystal. So that was very useful. All the structure that I show you are actually grown over here. So for protein crystallizations, it is important because a well-ordered crystal needed to detect the X-ray. So the X-ray diffraction data needed to solve the 3D structure. So crystallization with protein can be difficult because we don't know whether we can grow a crystal of the protein, right? Unless someone have done it. So otherwise, any of the new protein, we cannot tell or predict what condition that the protein will grow a crystal to be a crystal. So what in the crystal would be, if you look at the how the protein or molecule arranged in the crystal, it would be they will arrange and come to close together. In this case, most of the bonding would be hydrogen bonding, unless there's a disulfide bond formation between. But most of the time, or it would be hydrogen bonding, also a hydrophobic interaction, so that you move or when dissolved interaction, you move the molecule together, and they will arrange in a proper manner, systematic manner, that we could have a lattice. And in this case, then it will grow until it form crystal that we can see. Otherwise, if it's single protein, then we wouldn't able to see the molecule. And what is crucial for growing a protein crystal is that we need a pure homogeneous protein. And apart from that, crystallization also affect by the protein concentration. So we normally start with 5 mg per ml of the protein. And if you look at the PDB, or you look at the statistic in the PDB of how a concentration, the crystal grows, most of the time it will be around like 5 to 20, I think, of the 20 mg. So very few that you see that they are very low concentration, like 1 or 2 mg per ml. I think there are a couple of examples, but not common. Very high concentration also, I think, not very common, because many of the protein, when it go to very high concentration, they will tend to be precipitate. So there are environment or that protein are happy with, but generally that would be what from our experience. Yeah. So protein solubility is also important in the case that if its protein is not soluble, means that you are unable to have a high concentration. If you are too soluble, then probably you may have challenge in crystallizing it. And also it would be you need a lot of protein when it's very soluble, right? So that to later you will see what that it means. So the pH, the environment, the buffer itself also affecting the crystallization process. Temperature. So I mentioned the refrigerator or the incubator is about 20 degree. So one can set up the crystallization at 4 degree or 18 or to 20 degree. So depends on the protein. Sometimes it would crystallize at 4 degree, but not at 20, and vice versa. And so depends on the facilities that are available. If you have a cold room, so one can try different temperature. So those are the parameters that we use to grow or to screen if a crystal can form. Different type of solvent also may affect. Precipitant is really crucial. So in the commercial available kits that I mentioned just now, many of the conditions have a different concentration of precipitant and also different type of precipitants. Precipitant can be packed or salt. And Additional of the ions or ligand would be also help sometime if you know the protein would bind certain metals like zinc or magnesium. So the presence of those ions could be helping for the crystallization, or you know that the protein binds certain type of ligand. So I think a bit of ligand probably could help also sometimes to stabilize it, because we know that the binding of the protein and the ligand would affect the conformation, and it would be from if he's adding it would make it from disorder to order conformation, then it would help for the crystallization. So of course, the ionic strength also, the salt concentration, all are affecting crystallization process. So the protein crystal will form when protein concentration reach supersaturation. So it means the protein molecule in a solution are exceeding the solubility limit. So that's why what I mean here, if the protein is very soluble, then you need a very high concentration for the protein molecule to exceed the solubility limit. So this is a pickup from the literature here, where this depends on the method that used for crystallization. There are many kind of method that you can do crystallization. We generally use variable diffusion. So, but the process of getting the crystal would be from under saturation, which is very soluble. And if you go to super saturation, and with that, you may have a nucleation formation, and then you will see a precipitation. So the parameters that I mentioned just now here, it would affect the process of the path from under saturation to precipitation. So if you have, for example, if you have a protein, let's say 10 mg per mil, so you make the constant of your protein concentration. But if you add the concentration of precipitant from low to high, if you have very high concentration of the precipitant, let's say PEG in this case, polyethylene glycol, 3,350, let's say. If you have very high concentration, even 10 mg, you push the protein into the precipitation zone. But you have a lower concentration of PEG, then probably it will be still insoluble or super saturation or nucleation zone. So those are a parameter can normally we play with, so that we hope that the protein are in these zones and the crystal may form. So this is several major protein crystallization method from variable diffusion to microbatch or microdialysis. And if you see, this is a reservoir, this is reservoir. And the method that I mentioned we use generally is variable diffusion. And for the 96 well that I mentioned just now, it is called a sitting drop. In this case, the protein plus the reservoir, I told you, I mentioned 1:1 just now, right? Ratio 1:1 over here. So you have 1 microliter of protein, let's say 10 mg per mil, and you take 1 microliter of reservoir, you mix it over here, and then you will cover with a cover slip, and you will tight it, you will then seal it to make it vacuum, so that all the the condition over here that there's no air going out from the drop or no air coming in from outside. And in this case, since here will be more soluble in general, so the water will come to here, it will evaporate a bit, come here. And with that, the conditions, which is initially more soluble, suppose, if you then go through this process from under saturation to hopefully forming the crystal and to avoid precipitation. But sometimes we can learn. For the first couple of times, you may see if your drop is precipitate, then we may play around with the precipitants. So similarly, if we have from the sitting drop, which is small drop. Then if you want to move to 24 well, you'll be a hanging drop. In this case, the drop will be on the top of a cover slip. Similarly, 1-to-1. So in how long the crystal will come out, there's no answer for this. Depends on the protein. Sometimes you set up, immediately you see a crystal form, but most of the time you may incubate it at the 19 or 20 degree for a few days or even a weeks, then you will see the crystal forming. So in this case, this is a single crystal that we want to have a big crystal for X-ray diffraction. For micro ED, it could be slightly modified a bit of different strategy because you are asking, you would like to have a tiny, small, very small crystal. And so that would be a general process that we are applying here to obtain protein crystal. So as I mentioned, we don't have robot. If one have robot, you can just put a plate there, especially for this 96 well. So the machine will just take the reservoir and take your sample and the reservoir to put into the drop, and then you just can seal it. And for the imaging, we are using a light microscope, but for the laboratory have many samples and many groups of people working on protein crystallization. They may have this imaging system where like a hotel, where all the plate having a barcode, and if you then you can set the time, and the machine will then image it and record it in the computer, so you can see through the computer after the imaging system have imaged the drops. And they also can identify whether the drop having a crystal, and they may tell you. So with that, then you may not need to use your eye to look at the tray one by one or drop by drops. So also there are many facilities that are available. Actually, if one can, there's many samples available that you want to do it automatically. So it saves the time, of course, but it depends on the how many samples that we are having to be economic, right? So we always talk about the production and also the costs that involve. So there are limitations of crystallization. Those are some of the crystal that I grow or we grow over here. If the protein are non-soluble, that would be difficult to get into the crystallization process. If the protein have too many disorder region, so it's very hard to crystallize. And or the protein tends to aggregate. So when that tends to aggregate, so you couldn't arrange it in a proper manner. So you will just a climb or the protein that with different size or different different conformation. So that would be difficult to crystallize. And again, you may need to achieve certain concentration for the protein to be crystallized. So that would be the challenge, because some protein are not soluble. So even 5 mg per ml is hard to get. So those would be the challenge of the nature of the protein that we couldn't proceed with crystallization. So once we have the protein crystal, then we will then need to freeze it and send for X-ray diffraction. So in this case, if it's micro ED, you have the crystal, then you do the micro ED to do the data collection. Over here, we're using facilities from Malaysia Genome and Vaccine Institute or a Center of Chemical Biology, CCB for Ministry of Science Malaysia, or I also collaborate with my collaborator from Didcot, Oxfordshire, which is a diamond light source from UK. So if I have to send to UK, then we have to frozen the crystal and send it over. If it's for local, so I just put it into the car and we can bring the crystal to the X-ray diffraction screening and data collection. And here I show the facilities or the building that CCB, which is I collaborate with Dr. Teh from USM, MGVI, collaborate with Dr. Muhammad Anwar Jonit, and in Diamond Light Source, UK, with Dr. Yika Waterman. So for the single crystal that we obtain, we will put it over here under the X-ray diffractor. So in this case, we need a sufficient size of the crystal. At least, generally, we take it about 100 micron. Of course, if you send for a synchrotron, you could also use a smaller crystal. But for in-house X-ray like this, it's really hard to do with a small crystal like less than 100 micron. So I think for micro LED, I was told it's about 10 micron or 20 micron. Am I right, Prof. Tete? So it would be like 10 times smaller or 5 times smaller of the size. So in this case, we will then get either in-house X-ray or synchrotron. We'll get the X-ray diffraction pattern like this. When we see this, we normally will be very happy because it diffracts very well. Not all the case that you get a diffraction nicely like this. Most of the time, probably not. And so the in-house diffractor and synchrotron, that just to show the photo, just for you to have an idea what it looked like for those that never seen once. So this is a ring in the synchrotron. So where the electron will fly, and by bending it, you get the X-ray. So I have told you from here, recombinant protein, up to getting the X-ray diffraction, which is diffraction image. So you collect the data of X-ray diffraction. We normally call complete data, means that you have enough data that can determine the 3D structure. So the data collection strategy now is robotic putting the crystal, and also automated computational, where giving you a strategy how to collect the data. And once we obtain the X-ray diffraction data, we will then go to X-ray crystallography. That in this case, we will use a CCP4 package. So in the CCP4, there's many software that helped to process the data and also to determine the phase, solve the phase problem, to determine the 3D structure, to do the model buildings. So initially, if the phase is good, you may see a map like this. So when we want to know, we want to build amino acid insight into the electron density map in this case, then we will then see how the structure would look like after we build the model. So this is like you have a protein residue, amino acid residue, and here actually having a ligand. So those at electron density allow us to assign the molecule in the electron density, and you see the round here, it is a water, for example. So that would be atomic resolution for us to see the detail like this. So with this pipeline or this way, we solve the structure that I have shared just now, the few structures. Here, for example, the finding from the dust mite allergens. So actually, this is a dust mite allergen for this structure. And we would learn how the residue would serve as epitopes, for example. For the human protein, we see the detail of, for example, zinc ion binding to the human protein, and we can recognize a motif. For this hypothetical protein that I'm going to share in more detail, by solving this structure, analyzing it, we discovered it as a Nobel Cas2 or VAPD subfamily nucleus. So there are a lot of surprise after solving a structure, and we can learn a lot from a structure. either to answer the function or to know the function of particular residue in the protein. So now I give in more detail of this structure that we published last year, where hypothetical protein from Burkholderia Pseudomallei it is a gram-negative bacteria that it causing the disease called melioidosis. It's a very high fatality disease. The symptoms similar to tuberculosis or common pneumonia, and it is high risk for the people who are actually diabetes or having liver, renal disease, or several disease. And if you look at the distribution and also the cases that every year happen in the world, it's getting more alarming, especially when the climate change problem that we are facing now. So probably due to the high temperature, it would be expand further to the other part of the world. Otherwise, you will see there's endemic in tropical region, like in Malaysia and also Indonesia. So the bacteria also multi drug resistant, multi antibiotic resistant. There's no vaccine available. It's also classified as a bioterrorism agents. It could be a biological weapon. So people are concerned and would like to understand more about this bacterium. Although studies about the virulent factors and also the antibiotic resistance pathway has been described, but many regarding the bacteria still under study. If you look at the publication in the PubMed, it's only have a few thousands since 1948. If you just type Burkholderia Pseudomallei in PubMed, you only can find about 4,000 or 3,000 something of a paper. So it means that we still don't know very much about this bacteria. And if you look at the sequence that have been released since 2004, the genome sequence of the bacteria, Burkholderia Pseudomallei, which is including 5,800 genes. About 25% of the genes, encoded genes, were known as hypothetical at the time in 2004. And recently, we visit again the entire of the genome, and with the AlphaFold 3, we think our results show that probably by including all the structural information and the late updated sequence information, probably 2.5%, I think, remain hypothetical. Other of the genes would have some sort of information and can be annotated. We have submitted a paper under review for this. And by studying a hypothetical protein, we learned from one example about 10 years ago, I think, 13 years ago, a toxin that inhibits helicase activity of translation factor EIF4A, and the protein called Burkholderia lethal factor 1 now. And the paper was published in Science 13 years ago by reviewing the function, the structure and function of this hypothetical protein, which is a toxin that can inhibit the activity, helicase activity of translation factor EI4A, which is in ribosome. So our interest of investigating this for other hypothetical protein continue. So in order to understand the melioidosis pathogenesis and also whether those hypothetical protein are virulent factors or can be vaccine candidate, or are they important for bacterial survival? So those are questions that we ask when we are working on hypothetical protein. So this particular protein, PPSL1038, is a small protein, 88 amino acid, and is conserved in Burkholderia family, and also found in other bacteria genus actually. And through the literature, you can see that's all uncharacterized. No one knows the function of this protein. And there's no homologue in archaea or eukaryotic species. So we think that since it's conserved in most of, in a Burkholderia, say, family, it should be interesting protein to work with. And by look at the sequence, multiple sequence alignment, we can identify certain amino acids particularly conserved. And through the pipeline that I have shared just now, so we have gone through getting the genes. And in this case, we synthesize the gene from chemically synthesize the gene and clone into the expression vector, and gone through the protein purification, expression, purification, and crystallization process that I have shared just now. And we solved just for some detail of protein expression. So we can see that it is a soluble protein when compared with induced with IPTG. In this case, it's T7 expression system. Yeah. So we're using IPTG to induce overexpressions. So you can see the overexpression of the protein compared with uninduced. And it's also in soluble fraction. This is soluble fraction at 37 degrees. And from the heat trap affinity, because it has an N-terminal heat stack, and we could then purify the protein cleanly. And we subjected for side exclusion, and side exclusion not only can purify the protein, we also can learn the size of the protein from the detention volume. It came out, we can actually predict that the protein in the solution actually look like it's a dimer, because the protein itself is about 12 kilodalton, but when it go into the column side exclusion, it came out with a detention volume corresponding to about 24 kilodalton, suggests that it's probably a dimer in the solution. We know that protein can form oligomer, sometimes monomer, dimer, tetramer, and this can learn if we put it into the side exclusion chromatography. And the crystal defect very well. This is a crystal of BPSL1038 and to 1.5 angstrom for the native. For this one, actually is a ceramethionine label. Since if there's no structure available, in the past also we don't have AlphaFold, so we don't have a template to use for molecular replacement. In order to solve the phase problem, we need to use a heavy metal label. In this case, we use ceramethionine label to obtain the anomalous signal or data that allow us to solve the phase problem. So structure that we solve having a 2 subunit, which is in agreement with what we have seen in side exclusion, which is dimer. So this is a dimer of BPSL1038. And if you look at our structure, it's very charge negative. Only there's a few patches which is charge positive. From the 4 residue along the 88 residue, only 4 residue is positive charge, which is shown in the blue patch that you see just now. And by analyzing the monomer or the protomer of the dimer, we learn that it is very similar to Cas2 proteins. Here showing Cas2 protein from different type of CRISPR system. And this is 1038 compared to all others. This is VAPD protein, virulent associated factor protein. But other than that, those are all Cas2 protein from different type of CRISPR system. And you may ask how the AlphaFold would predict. This was AlphaFold prediction at the beginning when after AlphaFold came out, we use AlphaFold to predict the 1038 BPSL1038 structure, and this is the structure of it. And you can see that it looks different. There's very similar in certain part and significantly different in certain regions. So from the sequence alignment, we can tell that BPSL1038 is conserved in Burkholderia, and also similar to this virulent associated protein from this Helicobacter jejuli. This one. So we further compare the dimer or superimposed the BPSL1038 with the Cas2 dimer structure. You can see that they are really similar, right? And residue that known to be catalytic active residue in Cas2 is also conserved in BPSL1038. Furthermore, we knew that from the literature, and we learned that from other study, that some Cas2 protein can cleave DNA, some can cleave RNA, and for VAPD, it can cleave mRNA. So we look at the features and recognize that BPSL1038 having structure features that different than those of Cas2 nucleases that recognize the DNA or RNA. And we would like to further learn how BPSL1038 works. Would it work like Cas2, and would it cleave DNA or other substrate? So also for your information, if you're not aware, in the CRISPR-Cas system, Cas1 and Cas2 would form complexes that acquire the spacer. But during the spacer acquisitions, Cas1 is the one that would cleave the substrate, in this case, the DNA. But Cas2, it just provides as a scaffold for the Cas1 to dock, so that if you then bind to the DNA to cleave to acquire the spacer. So the nucleus activity of Cas2 is actually not acquired or not applied during the spacer acquisition in the CRISPR-Cas system. So what would be the function of Cas2 then, right? So we further look at the analyzing the BPSL1038. We found out that there's a this region in the putative catalytic active site, which is having a DSST forming around like a square, the sphere of a circle of active site. And if you searching this active site to all the structure that are available in the database, we couldn't find any similarity. So suggest that this is probably new type of motif that arrange from the structure that we solve from BPSL1038. Perhaps there are more structure that's sharing similar motif like this, but yet to be determined or reviewed. So we further analyze it, how similar of this active site compare with Cas2 structure. In this case, with the Enterococcus facialis Cas2, Cas1, Cas2 pre-spacer DNA complexes. So we superimpose to it and see that in the Cas1, Cas2 pre-spacer DNA complex of EFA, it shows that there are two magnesium atoms here, but in our case, we're having a water molecule over here. So, but in this case, it also suggests that if there's a substrate, perhaps magnesium can go in. So we then look further, possibility of the protein can able to bind the DNA. So we came out with the hypothesis that the R10 and R30 would actually clamp the DNA. So it like an arginine clamp to hold the DNA. So then the active site would then expose and to carry out the catalytic activity as such. The two aspartic residue would coordinate with a magnesium ion, which the water that coordinate with magnesium ion will be nucleophilic active in a way that the hydrophile group of the water will then initiate the attack to the phosphate group over here. So the transition stage will be either stabilized by arginine 30 together with the magnesium ion. So we went on to test whether this is true that the PBS1038 can cleave the DNA. So we test with the plasmid, in this case, PUP19 plasmid, and we can see that by incubating the plasmid with the protein, from the supercoil, you will go to the nick, and then you will see the linear species of the plasmid. So suggest that it is a endonucleases and it can cleave the DNA. So we also show that salt concentration would inhibit the activity, because when salt is 100 millimolar or 200 millimolar, you can see the activity is reducing compared to 5 to 50 millimolar. And we also found that pH 6 is most optimum for this activity. And interestingly, the divalent metal ion doesn't affect the activity. So by adding the EDDA, which is normally that would affect the divalent metal ion binding to the enzyme, but it didn't actually affect the activity or reduce the activity, suggests that the catalytic activity is not dependent on the metal ion. So we then ask how, if there's no magnesium involved, how then the protein or catalytic site would then able to cleave the DNA, in this case the PUP19 that we have seen. So we further model it and dock the DNA onto the protein and see that it is possible that the 2-cecil phosphate, which is in the minor groove of the DNA, which is about 12.5 angstrom apart, would be able to be docked on the top of this active site of that formed by the DSS T active sites over here, in which the R30 residue here probably would then interact with the DNA to hold the DNA over here and allow the active catalytic activity to be carried out by the active site like aspartic 11, serine 33. So we come with another proposal with that it could be similar to recombinase, where the serine 33 may initiate the nucleophilic attack instead of involving the divalent ion magnesium. And the R30 can stabilize the pentacovalent intermediate. So that's what we are proposing in our paper, after observing and looking at the structure and also the nucleus assay that we have done. So from the literature, we have learned that the aspartic 11 residue that equivalent to other class 2 aspartic residue over here is important for catalytic activity. And the mutation of residue that equivalent to the SST motif is also would reduce, in this case, those are ionase, class 2 ionase, their activity. So we also found the features of ST turn, which is unique in PBS1038, is not found in other class 2 protein. And likely that the DSST of motif is a novel motif that found from this protein. And we also went on to test whether really the protein are not able to bind the divalent ion. In this case, we soak with manganese, and we collect the data again, and we see that the water molecule remain a water molecule, is not a manganese. So telling us that even though you have high concentration of manganese, it didn't actually occupy the position that we think can be occupied by the magnesium. We also went on to study the function of this protein to see whether it's a toxin. And by fitting the protein, or either the protein itself or the E. coli that contain overexpressed recombinant protein, we found that it's actually not particularly toxic to the C. elegans. So look like it's not a toxin. And the C. elegans also live very well, even though eating the E. coli with this recombinant protein. And we also test with human breast carcinoma cells using a cell line, and to test whether it is toxic to the cancer cell line. It's also found that it's not toxic to the cancer cell line. Look like it is not toxin from this data to show. So from the works of this work, we found that BBSL1038 share a common ancestor with a Cas2 and a VAPD protein. So it's likely a new subclass similar to Cas2 and VAPD. Overall, a negative charge and having nucleus activity, and surprisingly with a metal-independent manner when it's cleaving the DNA, the substrate, and having a unique functional site, but the physiological function yet to be determined. So there are more questions after we have finding some new information. With that, we are being asked also during the review process what kind of function it would be. And we are recently getting a grant to further study the physical the function in terms of more substrate to be tested. For example, single stranded DNA, RNA, and also physiological function of it. So that's what we are going to continue with this project. And I think I forgot to mention, in the BP, there's no CRISPR-Cas system has been reported. So it is not a Cas2 protein likely. If there is, it means that there is probably a very different sequence Cas protein that would exist in BP. But so far, there's no report about CRISPR-Cas system in BP. This is the first protein, Cas2 protein, that's similar to CRISPR-Cas protein in BP. So there's more to be reviewed or to be investigated. So with that, I would like to thank for this particular work, Sophia, who grown the crystal, Shahila, involving in characterizing the function of the protein, and also many other of the work by these two master students, very dedicated, very good students working on this difficult protein actually. And many peoples that involving over here, I don't name them, but all of them contribution are very important. The funding is from our university, University Kebangsaan Malaysia, and also Ministry of Higher Education, and with collaboration of multiple institutions, USM, UPM, University Malaya, SCCP from USM I mentioned, MGVI, Diamond Light Source from UK. And some of the work that I show you, the structure that we have solved, also work together with many collaborators from different universities. And with that, I thanks for your attention. Thank you.
Okay, thank you very much, Professor Leong, for a very complete and very nice and interesting presentation. Yeah, you explained about how the basic information about the purification, also crystallization is very good information for us, especially for all the audience here. And also you also introduced us about your research paper that has been published in 2023 last year, in Nature Communication. I think this is very nice information for us. Before I start the question, I think there are two questions in the chat. Yeah, and also I invite all the audience here. You can raise your hand and open your mic, and also you can write the question in the chat. Yeah. So Okay, I will read the question from Dr. Marisa. Yeah.
Right.
She asked about the how to crystallization of their protein. The size
Mhm.
is 14.5 kilodalton, and what have to be concerned, yeah, to avoid the protein lysis during the crystallization process. For your information, Prof. Leong, I
Mhm.
think we are now struggling to obtain protein for the micro-ED, using the cryo-EM. So protein under less than 80 kilodalton, we try to make a crystallization. But as you already mentioned, that the micro-ED needs
Mm.
only very small size of the protein,
Yeah.
uh, like point two, maybe below 0.2 nanometers. Yeah, below 0.2, below 0.2 micrometers. Yeah.
All right. Right.
Yeah, I think it is also good information if you can share us about how to prepare the protein crystallization, just a small size. Yeah, because usually for the crystallography you have to make a bigger, large crystal, but for the micro ED we only need very small crystal. And how we can provide this small crystal using this technique, please. Oh,
Right. So I'm as I mentioned just now, maybe I stop sharing, yeah. Right. So I mentioned the for micro ED from the discussion also with participants in the ASCA, the conference in Kuala Lumpur last week, Asia Crystallography Conference. So some participants, they were working on micro ED. So when during the discussion, I realized that there's something like a bit reverse, because normally for X-ray people, we would like to grow big crystal. We would like to have a single big crystal. And when I realized that the big crystal is not good for micro ED, then it came to uh, the mind or the discussion that that would be also a challenge, right? Because the challenge is over here where when you grow a big crystal, we can see under microscope, light microscope. So like 50 micron, I think you can see it already. Smaller than that, you can see maybe a tiny, you can see something. But if it's too small, it's actually very hard to see, right? So to me, to be honest, I don't have experience in growing very small crystal, because normally when we see some heat, oh, there's look like there's something, then we will then optimize to make it big. But now for micro ED, that would be already good, because when you see something, then you say, okay, now we can then use this for preparing the sample, right? For micro ED. So I think in this case, perhaps from my personal opinion, yeah, if we do know, if you do the crystal screening, you have a big crystal, then perhaps you would like to de-optimize. But optimize is to make it big. In this case, you may want to apply the condition to let the crystal not growing too big. So instead of making it better, you may want to make the condition a bit worse, right? So that the crystal may form a nuclei or small, and that's enough for screening. But I would say that if I don't have any experience yet, I perhaps need to learn from people who are doing micro ED. But from my crystallization experience, I would likely to if I know the condition, then if I know that 10 meg, let's say, would give a big protein, then probably I will go down, perhaps. Instead of optimizing it to bigger, I will go down to like maybe 6 meg or so. So likely that if I see the crystal didn't really form, but I knew the condition is allowing the protein to form, I will work out from there to get a small crystal. You get what I mean? So for the crystallization, I think is that first, the protein have to be able to come and form together, crystalline, right? Second, for single crystal like me, I would want to optimize it to not only to form crystalline, but form big, right? But in this case, you don't want to have big, or we don't want to have big. Then you don't want to optimize. So from the condition that help to optimize actually can be used to not optimize it. That's what I think for the big crystal. But I think it need to be practical. I don't have a, but we can try. We can also learn from people who are doing it from the micro ED now.
I see. Thank
Okay,
you,
I
Prof.
see.
Dr.
Yeah.
Marisa, do you have any?
Yes, yes, thank you, Prof. Prof. Chenglong and Dede. Actually, we are have a protein that will be analyzed on cryo-EM, but the problem is the protein is very small. So some college suggest to do crystallize. But when I heard your presentation today and your answer on my question, I think you don't suggest to do micro ED in crystallization of our protein.
No, I'm not particularly addressing that protein, Melissa, yeah.
Yes, Marisa.
Marisa.
So no, just now I didn't particularly address to your protein particularly. So for may I learn your protein is 14.5 kilodalton you mentioned?
Yes.
Yeah, if it's soluble protein, you can do crystallization. Yes, no problem.
So the requirement from crystallization is soluble or not, isn't it?
Soluble, first soluble, yes.
Yes, soluble.
Homogeneous, then
Yeah.
homogeneous.
Yes. Okay. Is the requirement, yeah? Soluble and homogeneous, yeah.
Yes, then you perhaps concentrate to at least 5 mg per mil before you try to crystallization. Start
Yes.
crystallization at least. If it 10 mg per mil will be good. Normally what we do is this. If we achieve 10 mg per mil of the protein, we will do a screening. And in the screening, under the microscope, you will observe the condition that you put up, let's say 96 drops.
Yes.
Is that all the drops are soluble, most of the drops are precipitated. And if it's 50-50, it means I think you are in the right concentration, because if most of the drops are precipitated, it means that even low concentration of the protein is tend to be precipitated.
Okay.
If most of the drops are soluble, it means that you may need higher concentration. Because if you remember the graph,
Yeah.
so from soluble to precipitation, right?
Yeah.
So if you 10 mg per mil, most of the conditions still soluble, then you may increase concentration so that you push it to the supersaturation or the nucleation formation.
Yeah. Okay, Prof. Thank you for the suggestion, Prof.
Thank you.
Thank you, Professor Leong and also Dr. Marisa. Okay, I will invite Nadia. If you want to open mic, please.
It's also our
Nadia.
team, Pak Dede.
Oh, okay, same. Okay, thank you. So the other question is from Herdayanto. If you want to open mic, please.
Hello, good morning.
Hello,
Yes,
good morning.
Wait, let me. I cannot see myself. Sorry, I have some trouble with mine. Let me change it. Okay.
Hello.
Right. So thank you. Yes, I would like to ask about working with protein refinement, and then because I would like to know whether the refinement is good enough or it's not good enough to submit for PDB, and if I find some of the residues, it's really hard to move to the green area in Ramachandran plot, I mean, it keeps moving on the red, and when I try to change it, I mean, it changed the others as well. And do you have any suggestion or maybe any tips or tricks to work with that? Thank you.
Thank you. Yeah, that's always tricky, during the refinement. So for audience, maybe for modeling, normally when we are trying to model the residue into the density map, so when is the time say that we are stopping? So the question from Hidayanto, right, is when we think that it is good enough and we stop, right? So yes, always at the time, for me, normally is when I do the refinement or I do the model building after refinement, the moment that I stop normally is when I built whatever that I can build, I already built. And if I have some density that I'm not sure what it is, and if I put something in, if I think it is misleading, then I will not put. If I think after I put and it still didn't improve, or sometime actually, so sometime I think most of the case is the density is not clear, so we are not sure whether we should put anything in. So that if I'm not sure, I will leave it out normally. If it's water, then normally we know that it should have interacting with residues, so we can put water. For particular residue that you see there's a density, but you cannot fit in, right? Normally I will try whether I take it out or I take a few residue out and try to fit again. And if it depends on the program that you use, I use RefMark 5. I'm not sure which program that you use for refinement, and I use CUT. So I use CUT to model, and if the density is really confident, even though it's out from the Namatrandan out layer, and if you can explain that this fitting is correct and probably is important, sometimes it may outlayer a bit from the Namatrandan. Not very common, but occasionally, I think if that is the case, I think it's all right, because people would see that that's actually the density, right? So if the resolution is high enough, then I will leave it. But if it's less, the resolution is not very high, then I think we may not think that what is wrong is actually right. So I think first I will consider the resolution. If the resolution is very high and I know that the red atom should be there, but sometimes Namatrandan may not fit, then I probably will take it because I will believe in the data, right? The experimental data that I'm confident with. But if the resolution is not high, the map is a bit bulky, that you cannot really confident the atoms or the residue should be, then I would think the model is wrong, and I will try again to fit it. That's my take, Dr. Hidayanto.
Okay, thank you so much. Thank you.
It's very tricky. Every part is different, right?
Yeah, I'm trying to fit with the R3 as well. And thank you.
All right.
I will try it.
Good luck.
Yes, thank you so much.
Okay, thank you, Pak Herdayanto, for the question, and also for Prof. Leong. Thank you for the answer. And then I invite also Ahmad Fadullah Husaini. Do you want to open mic?
Okay, thank you so much, Prof. Dede and Prof. Leong, for your presentation. I do have a question for you, Professor. So, considering we now have methods to predict high quality predicted structure for novel protein using AlphaFold,
Yeah.
and some research also found that it can be used as a search model. But as shown in your case, the
Yes.
predicted structure can be quite different from the actual elucidated structure.
Yes.
So, do you have any suggestion or maybe tips or trick to improve the predicted structure before being used as such model for molecular replacement?
Yes. Okay. Maybe in this particular case, I share with you. So I show you the monomer look a bit different, right? So when the dimer, when we predict, it's also look a bit different. So normally in this AlphaFold, at the beginning, we obtain 5 structure. So we have looked at all the 5 structure that came out from the AlphaFold. And actually, the structure number 3, if not mistaken, is actually alike like our structure, the structure that experimenter determined. So it means that it's really powerful, yeah? I see it myself where our structure haven't been deposited, but we use AlphaFold and it predict the dimer and it give you 5 dimer. And number 1, number 2 is not a bit of incorrect because of the beta sheets arrangement. Number 3 is alike. So the question here is, Ahmad, how if we don't have experimental data, number 1 question is hypothetical protein. How do we know the protein is actually dimer or trimer? So if we don't have this information, then you may put this as one of the parameter, right? When we predict the structure, you may want to predict monomer, dimer, tetramer, or so on. And when you in this particular case, I explain a little bit more detail. In this particular case, if I knew that I don't have structure, let's say, but I have a protein that I know is a dimer, I first predict the monomer, and I found out that it's very similar to Cas2, right? And the Cas2 protein, most of the time, are dimer. And I would think that this structure would also form dimer. And if my experiment data told me that it is likely a dimer, then I knew that this structure, like Cas2, it would form dimer. And I will predict it as a dimer. And from the 5 dimer that I predict, I would then further analyze each of the dimer. During the dimer prediction, you can then break it down, each of the protomer from this dimer, and reanalyze each of the monomer and see whether they are similar to Cas2. The one that's similar, in that case, then probably I will take it. And this probably will allow me to solve the structure using AlphaFold, identify that the number 3 is probably the correct structure instead of number 1 and 2. I don't know whether I explained it clearly.
Okay, thank you so much. I understand what you said. So we need to compare it with other known structure or a protein in the same family to predict whether the predicted structure is correct or not.
Yes, that information will be very important for making decision.
Okay, okay. Thank you so much for the presentation.
Thank
once
you.
again. Okay.
Okay, thank you very much for the question and also the answer from Prof. Leong. So, Prof. Leong, I have a question about your research published in Communication Biology in Nature. So,
Right.
about the protein BVSL1038, you already revealed the structure, and you said that the structure of the protein is almost high similarity with the Cas2 in CRISPR and also VAPD for the virulence associated protein, right? So you already feel this protein, and then my question is, what is the whole function of this protein in this bacteria? Is it also it has a like a virulence activity for this bacteria that infected the human, or because also you said that this protein also has the good toxicity for the MCF7, right, at breast cancer for human. So
No toxicity, yeah.
no toxicity.
No.
Okay, could you explain about the whole function of this protein in this bacteria?
Right. So we are still searching for the function, but in our mind, so it is similar to VAPD, which is a virulent associated factor, right? Protein, and similar to Cas2. So some Cas2 that show that they would have a virulent factor, it could be. But in our two experiments, we show that it's not toxic to the C. elegans, not toxic to the human cell line, human cancer cell line. But of course, it depends on the assay that we did, right? And during the reviewing process, one of the things that we think of, if it is similar to CRISPR, we should have other CRISPR protein. Then it would be like a Cas2. Since no other CRISPR protein found in the genome, we know CRISPR protein is the immune system for the bacteria, right? And would this protein part of the similar system of CRISPR, which is not CRISPR but similar, and it would function also towards immunoprotection of the bacteria. It is possible. We are hypothesizing it's possible because we learned that Burkholderia Pseudomallei have less bacterial phage that can infect them. So the only way to test this is probably to do the knockout or knockdown of the Burkholderia Pseudomallei and then challenge with the bacterial phage. That's number one. Second, we are also planning would the DNA is actually the subset of it, or actually if it's DNA, are there a preference DNA? Is that virus DNA would be cleaved better, or RNA would be cleaved better by the protein? So we are at this line also investigating, because we have seen from our assay, we didn't report it in the paper, is that if you use PCR product, it seems cleave faster. If you use a plasmid that extracted from bacteria, it cleave, but seems like slower. So we also not really understand what is happening. So if we could address this, then I think you provide better information for us to derive their physiological function. So we still in the line that probably is immune system.
Okay, yes.
But virulent factors possible, but from our assay so far is not supportive towards this line. But we cannot deny that our assay is not fit to what it should execute, right? The function of the protein. Yeah.
So in the future, you would like to explore more about the nucleus activity of this protein, right?
Yes.
Yeah.
As well as if possible, we can do challenge with bacterial phage. That would be have to collaborate with another researcher lah, that they can grow the BP. In our lab, we cannot grow the BP.
Yes. Okay. Thank you very much, Professor
Thank you.
Leong. Oh yeah. Okay. We have a question from Dr. Adrian. Dr. Adrian now is also joined with us from NICE. Thank you,
Hi.
Dr. Adrian, for joining this webinar. Yeah. Hopefully next session we will invite you again. Dr. Adrian already become a speaker in this webinar. Yeah. Please,
Thank you.
Dr. Adrian.
First, just thanks. I would like to thank the organizer and also allow me to join this webinar. I'm very excited when I saw Prof. Ng is going to present. So, Prof. Ng, I'm very amazed by your research on discovery the function of the novel enzymes in the gene by looking at the genome. I just have a question on the, is there any meaning, like, because in the genome is sandwiched between the two component system and also the ABC transporter. So, and also I think TCS component is also important for biofilm formation in BP. So is there any relation or in the role that located at this particular locus in the genome?
Yes, thank you, Adrian, for the questions. Yes, we actually had looked at it, the location of the BP1038, BPSL1038, and also if you see the slide that I showed, there's BPSL1038A
Yeah,
always together, and
yes.
there's ABC transporter, right?
Yeah.
Yeah. So we were discussing about this, and we have done the works to see whether BP1038 and BP1038A can be a TS system. And unfortunately, we never able to produce BP1038A. This is the work from Shahila. So we have tried many attempts to overexpress the BPSL1038A, but we are never able to get the protein. And I think we also call express once. We also not able to get the complex protein.
I see.
So we are not, and the BPS1038A, when you predict with AlphaFold or like StarFold, it's just a disorder protein. So it's not a fold protein. And I think we also try to make using put the two gene and predict, and we also couldn't find the complexes. So we don't have an answer for whether they are TS system in this case yet. Yeah. I mean, the BPSL1038A is an antitoxin. We don't have question for this.
I see.
But for the ABC transporter side, this is interesting because we knew there are some enzyme could be in between the membrane that could cleave nucleic acid, right? Or protein. So whether it is involving in biofilm formation, where we knew that BP form biofilm, BP does form biofilm, and also in order to process a component of biofilm that needed to form a biofilm like polysaccharides, nucleic acid is one of the major components of biofilm formation. Whether it is involving in this, we don't have answer yet, Edwin.
Oh, I see.
But it is interesting that we did discuss actually quite deeply on would the enzyme somewhere located in the membrane, since it's so well conserved of the APC transporter and also BPSL1038 and 1038A. But we don't have a solid answer for this question yet. No.
I see.
But we
Thank you very much.
we
Yeah.
have several thought of it. Yes.
Yeah, very interesting. Is it okay for me to ask more question, or the time is up? Or
Yeah, please, Dr. Aditya.
yeah. So I also interested on the structures. You mentioned the serine might be the other nucleophile. So I just wonder, because serines normally is quite stable, say you need an activator like the catalytic triad. So in your case, because there is no metal, so how can it be activate enough to start the nucleophilic attack? Is there any is it water or someone to help him to activate the serine?
Yes, Adrian, I thank you so much for this question. Very challenging one. If you look at my paper, our paper, so we didn't put it very solidly because using serine as a to initiate the nucleophilic attack is not very common if you look at the literature. From our paper, we cite the recombinase. So the recombinase example with serine that initiate the nucleophilic attack for the DNA. But throughout the literature, most of the nucleases, they are mostly with divalent ion, either 1 or 2, to initiate this attack. So since our assay show that divalent ion seems not involved when we add the EDTA in, or adding or not adding the magnesium ion, we see similar activity. And for your information, the assay that I show, the protein is actually 3-step purification, which affinity, and then ion exchange, and side exclusion. Why is that? For the protein crystal is 2-step. For the assay, we're using 3-step. Because we are worried that just 2-step, this is a nucleus, so you may have other things still bind, and you may have, and to be honest also, where we found the reproducibility problem using 2-step protein purification sample. And hence, we reproduce the protein, and it actually took us almost 1 year to revise the paper, because we have to re-establish the method to purify the protein, which if a nucleases or protein that bind DNA, you may want to have a high salt in the environment in order to wash out anything that bind to the protein. In this case, perhaps nucleotide and things like this. So we expose to high salt, and we try to make sure that there's no other things apart from the protein before we conduct the assay. So with that sample, and we found adding magnesium or without not helping, and with the EDTA is not inhibiting, and we propose that it's maybe not a divalent ion dependent. Initially, we proposed divalent ion dependent because most of the literature, if you see the nucleus, they would have magnesium ion. It's 1 or 2. And without, how could I support? So I went to literature and searched any of the enzyme that cleave without divalent. So there's a few examples from the combination. So that's how we learn from there to propose this. How actually it would happen, I don't know, Adrian.
Okay.
We would have to investigate.
More questions
Yes.
came from that.
It's interesting.
From this research.
It's interesting biochemistry here.
Thank you very much for the answer.
Thank you.
Yeah. Can I have last question? Is it
Okay,
possible?
please, Dr. Adian.
Okay. Just now, since Prof, you mentioned about the PCR product, the difference between PCR product and the plasmid from the E. coli. So is it possible to relate to the methylation of the DNA in E. coli, or is this something like that?
Yes. Thank you, Adrian, for this. Yes, we also think in the same line on this, but we didn't have evidence to prove this. So in order to prove this, probably we need to order a proper sample to test with and without methylation and see same sequence. Only then we can propose. But from our observation and informal discussion, that's what we think that perhaps the DNA that we obtain from the cells, extract from cells, may have modification that maybe delay the process. But we don't have any evidence to say.
Okay.
So
understand.
I cannot strongly suggest this apart from unless we have evidence.
Okay. Thank you very much. I think that's all from me. Sorry to ask so many questions.
Thank you, Adrian.
Thank you very much for the presentation.
Okay, thank you very much, Dr. Adian, for the question. Yeah, I think we are now already in the end of our webinar, Professor Leong also. I think this is very interesting discussion about the very basic information about the purification and also crystallization, also information about the protein you already make the recombinant from the BV. So actually, I have so many questions about this topic, but because of time, we may later we will discuss more detail about the purification, crystallization. As also, I would like to invite you also come to BRIN, and we have a
Thank you.
more collaboration also. Yeah. Also, I think all this audience also as a PI of the structural biology grant. So I think we will make a like a future collaboration from between BRIN and also UKM, your university. Also, yeah, we would like to invite you to come to BRIN.
Thank
Okay.
you.
Thank you very
Thank you
much.
so much. Thank you so much for that, Prof. Dede. Yes, looking forward to work together with you and with others in BRIN.
Yes, thank you. And okay, I would like to give back to Mbak Yati. Thank you very much for all.
Thank you, Prof. Dede.
Thank you, Dr. Dede Heriulyanto, for leading the discussion, and thank you, Professor Chen Leng, for the interesting presentation. Ladies and gentlemen,
Thank you,
we have
Yati.
finally come to the end part of this webinar. I'm Yati Listiani. On behalf of the committee, I would like to say thank you very much to all participants and see you in the next event. Assalamualaikum warahmatullahi wabarakatuh. Thank you.
Bye.
Thank you, Prof.
Thank you.
Pak Dede.
Thank you.
Adrian also.
Thank you, everyone.
Thank you, yeah.
Thank
Pak Albi, thank you so much.
Thank you, Prof.
Thank
Abe.
you. Thank you,
Yeah.
Prof. Dia, Prof. Chen
Okay,
Leng.
Prof, I would like to invite you.
See you.
Thank you. Bye-bye.
Bye bye for now. Bye bye. Thank you.
Bye.